Everybody, I welcome you all back, and today's topic is gymnosperms. And today one more class, BSc 2, is joining us, right? I already told you that these topics are not for particular class, they're for all classes. If we are supposed to clear IAS, IPS, Indian Forest Services, UGC, OCT, the basic knowledge is required, right? So everybody, including MSc, gymnosperms is also in MSc. So you should learn it very carefully, and this topic is dealt particularly keeping in mind the objective questions, right? So stay indoors, stay safe. I will be back. Stay on. Good morning, everybody. I welcome you all back. And today we are dealing with the gymnosperms. As gymnosperms is the word which is used in 300 BC by Theophrastus, a pupil of Aristotle, in his book Inquiry into Plants. Gymnosperms as made up of two words, gymnos means naked and sperma means seeds. Gymnosperms are bridge between pteridophytes and angiosperms, hence they are called phanerogams without ovary by Goebel. And the word tallest gymnosperm is long-lived plants, 4,000 years, and height of 100 meters is Sequoia gigantea. And in contrast, there is the smallest gymnosperm, Zamia pygmea. It is 4-5 centimeter long only. Most of the gymnosperm shows xerophytic character, sunken stomata. Because of time constraint, I just tried. Others are the transmission tissue, which I discussed later on. So I am coming to my topic, that is cycas. Cycas is originated towards the end of Carboniferous period and formed dominant vegetation during Triassic of Mesozoic era. This period is called Age of Cycads. It's very important. The plant body is sporophytic. It is differentiated into the root, stem, and leaves. And the other part, that is anatomy, and other part I will deal later. So I am directly coming to the reproduction. Reproduction is the first type is vegetative, and it is in this bulbil is the common method, and bulbils they develop from the axil of scale leaves. We have two types of leaves, that is foliage and scale leaves. They are oval structure with broad base and tapering ends. And these bulbils on detachment form roots towards the lower side. Male plant bulbil, they give it male plant, and female give female. So hence cycas is strictly dioecious. The second type is by the suckers which develop from the roots, that is cycas circinalis. You will see the bulbils. This is oval structure, broad at the base and pointed towards the tip, and then it gives rise to a young leaf. So now we are coming to the second part, that is the sexual reproduction. The cycas is heterosporous and dioecious. In India, most of the plants of Cycas circinalis, this is very important, are male, and those of Cycas revolta are female. So now we are coming to the male cone, which we also call strobilus. The male cone are born singly and terminally on the main stem, right, like this. The apical growth of stem is continued by the formation of axillary bud at the base. Axillary bud will form, and apical growth will continue and push this on this side, right? It becomes new stem apex. As it grows, the male cone is displaced. I just told you, displaced towards one side, and to one side. The stem of male plant of cycas is therefore a sympodium. This is very important, because when later on I will talk about the female, I will say monopodium. So this question will come. See, first the male cone is formed at terminal, and then axillary bud, it will move up and it push it to the side, and then again same thing happen, and this structure is called sympodium, right? So next we are coming to the structure of male cone. The male cone is compact, woody, fusiform or spindle shaped with 20-60 centimeter long. In Cycas circinalis, male cone is about 80 centimeter. This is same Cycas circinalis which give rise to the male plants. I already told, right? It bears a microsporophyll which are arranged in, very important here, acropetal succession on the central axis, youngest at the top and oldest at the base. The microsporophyll of middle part are the longest. See, this is the cone I make, so made for you, and here is a cone axis, and this is microsporophyll. In books it is wrongly made. It's very easy to make it. Just when you make, then don't lift the pencil. See, 1, 2, 1, 2, 1, 2. See, this stroke will go up and this will go inside. This will go up and this. In books you will see like this much distance is there. So if this much distance is there, then because of dehydration, these spores, they will dehisce earlier before they mature. It is same like you can ask 3-year-old kid and take this suitcase and go to the office. But it's not possible him to reach because he's immature. So this is the right form which I told you. In books it is not given. You will see it is a central axis and microsporophyll, they are arranged right angle to it, and here these microsporangia, they are covered by downward projection and upward, so that there is no evaporation and they will not dehisce, right? So now we are coming to the next part, that is detail of microsporophyll. Microsporophyll arises as a small outgrowth right angle to the central axis. I just told you. Microsporophyll is a woody flattened wedge-shaped structure. Each microsporophyll contains two surfaces, adaxial, which is expanded sterile disc called apophysis, and the abaxial fertile part consists of thousands of microsporangia which are arranged in sorus. See, this is microsporophyll and this is the sterile part called apophysis, and this is fertile part in which their microsporangia are arranged, and when a lot of microsporangia are arranged, it is called sorus, right? So then this microsporangia produce microspores. So microspores are basically the pollen grains is rounded uninucleate structure surrounded by outer exine and inner intine. So when microspore will come, it is the first cell of male gametophyte. Now we will start the production of male gametophyte. So because in books it is given microsporophyll, but in continuation we will go like this, which I am telling you, right? It's easy to understand. The microspore start germination inside the microsporangia. That is why this germination is called in situ. Same, right? In case of megasporangium also, the first division is transverse. This is very important. It produces two unequal cells, small prothelial cell and large N3D cells. These prothelial cells lies towards the proximal. Suppose this is microspore, and this prothelial cell will come here, and this will called N3D cell. And the N3D cell divides into two again, distal tube cell and generative cell. So it will form two again. This is distal tube cell and generative cell. And then this time the microspore is at 3-cell stage. So this question will come. Pollination occurs when microspore is at the 3-cell stage, which is liberated and are carried by wind to the ovule, right? So this is basically the structure. I show you. This is the megasporophyll I made for you, right? A bit change I told you, which you have to manipulate, right? This is the microsporophyll I told you, right? So now we are coming to the male gametophyte. See, here the first division, it is prothelial cell and N3D cell. And N3D cell, they divide again to form this tube cell and generative cell. This is the 3-cell stage at where the basically pollination occurs, right? Again, Again, we are coming back to our structure, that is the development of male gametophyte after pollination. After a period of about 4 months, inside the pollen chamber, the generative cell divides. A pollen chamber, I will let you know after some time. The generative cell divides to form a stalk cell and body cell, right? The stalk cell is near the prothelial cell. Like the structure we are making here, it again divided into the stalk cell. Stalk cell will come here, right? And body cell. And the body cell, stalk cell is near the prothelial cell and does not divide further. The body cell divides to form a 2 sperms, and the sperms are very characteristic feature. So these sperms are like lattu, which Beyblade, we said we just circulate when we are kids. Top shaped sperm is a 180-210 μm. It is a motile structure, clearly visible to the naked eye. This is the largest in the plant kingdom, and it has the 5 spiral bands left to right with cilia. And this swimming habit of cyclic sperm is a primitive character. Right? So see, this is the structure of antherozoid, where it is like a lattu, I say, and having a cilias, spiral bands left to right, and these are easily visible to the naked eye, right? So now we are moving to the next part, that is the female reproductive organs. And in female reproductive organs, first we do the strobulus, ovate strobulus. So we are talking about the female strobulus, and the true female cone is absent in cycas, and the female reproductive organs are present in the form of megasporophyll. Megasporophyll, they occur in these megasporophyll, they occur in the closed spirals in acropetal succession, same as that we did earlier, at the apex of stem. They are arranged like an ordinary crown of leaves and collectively called female strobulus. The growing point of the stem remains unaffected by their development and continues its growth directly through the loose strobulus. Like in case of male, the axillary bud will come and take the role. Here it will continue to grow. The stem of female is thus monopodial, in contrast to the male, which is sympodial. Right? Now we are coming to the structure of megasporophyll. Each megasporophyll is considered modification of foliage leaves, and it is differentiated into 3 parts. That is the proximal stalk-like axis, middle part having 2 rows of oppositely or alternately arranged ovules, and the distal expanded dorsiventral flattened sterile part. Right? First we will do the cycas circinalis, and these all megasporophyll, they vary with their shapes of their sterile part and the ovules. Here the shape of that sterile part is ovate to lanceolate with long acuminate point. Right? See. See. See, this is circinalis, and we have long acuminate tip, and the ovules are, we have 3 plus 3. Like revolta, we have second is the revolta. Here the ovules are 2 plus 2. Right? So we are coming next to, then is the revoluta. The megasporophylls are covered with yellow or brown woolly hairs, and then we have Batomy. They are also again same like this of circinalis. They are ovate to lanceolate, tapers into long acuminate, and 2 ovules on the other side. But these ovules, they are alternate. See. This is Batomy, and the ovules are alternate. Right? Then this is Pectinata. We have woolly growth, and Siamensis, right? So we are again coming back. That is, we have the next structure is we have pectinata. Is a small marginal teeth blade, orbicular, long, acuminate with pectinate, which I showed you right now, right? So this is the structure of megasporal we discussed, and now we are coming to the next, that is the structure of ovule or megaspongium, right? Cycas ovule is orthotropous, unitegmic, short stalk, seen with naked eye. A single integument is very thick and covers the ovule from all sides except the mouth, called micropyle. The integuments are 3 layers. Outer green fleshy layer called sarcotexta. This is sarcotexta. And the middle is sclero. If some have some problem, so SS is same. You will do ABC. A will come first and C will come later. But you have to remember that the outer layer is sarco and the middle is sclero and the inner fleshy layer. The tissue encased in the integument is called nucellus. The apex of nucellus grows into a beak-like outgrowth called nucellus beak, which projects into the micropyle, right? Some of the cells of the nucellar beak disorganized to form hollow cavity called pollen chamber, right? Within the nucleus lies enlarged megaspore or the embryo sac, right? Before going to the, I will show you the structure, and here we have the first is the sarcotesta, then sclerotesta, and that inner fleshy layer, right? So then we have this nucellus, and then it projects in, it is called micropylar chamber, and this disintegration of these cells result in formation of this pollen chamber, and these are the archegonia, right? So when we see this structure, this is very easy: outer fleshy, middle stony, inner fleshy, and this is the pollen chamber. And from here, this is nucellus tissue, some cell disintegrate, and these pollen tubes, which I just told you, they hang into this chamber, right? And these are the archegonia, right? So next we are coming to the female gametophyte. In case of female gametophyte, one of the cells, they become increase in size, dense in cytoplasm, and it start behaving as a megaspore initial cell, right? And these which are also called megaspore mother cell, and it divides by meiosis to 4, and 3 they becomes disintegrate and 1 remain functional. So megaspore is the first cell of female gametophyte, and megaspore also develops in situ as we did in the microspore. The functional megaspore increase in size, I already told you, and then what will happen, a vacuole appears in the center, and the nuclei, like this is megaspore, vacuole will come and the nuclei will move towards the periphery, and wall formation will take place from periphery, and the wall formation start from periphery to inside, and this is called centripetal method. This haploid tissue is called female gametophyte. After the formation of female gametophyte, the cell at the micropylar end enlarge and form the archegonial initial. See, this is the MMC, and it divides meiosis to form 4, 3 degenerate and 1 remain functional, and this functional will divide again by mitosis to 2 and then 4, 6, 8 and further to many nuclei, and then the vacuole will appear and it pushes the nuclei towards the periphery, and the cell division will start from periphery towards the inside, and we will get this gametophyte, female gametophyte. So one of the cells, it start behaving as the archegonial initial, right? And these archegonial initial, which I told you, and this is archegonial initial, and this archegonial initial divided into the two, that is primary neck cell and central cell, and the central cell again divides into two, that is the ventral canal cell and egg, right? So this because in this there's no neck canal cells, which we did in the bryophytes also. So see, archegonial initials, it divides by transfer division to primary neck cells and central cell, I showed you just now, and it undergo vertical division to form two-celled neck and central cell to form ventral canal cell and egg nucleus, and there is no NCC. We can NCC called neck canal cell in cycas, which we did in the earlier class, that is bryophytes. So egg of cycas is the largest of the plant, and cycas hircinalis, which I already told you, they give rise to male plants, they having a 0.5 mm of diameter. So next, the fertilization is siphonogamous. It's very important. When the pollen tube acts as a hostorium and as a sperm carrier, that is called siphonogamous. And here the diploid zygote, which is the first cell of sporophytic generation, right? I already told you the first cell of gametophytic generation is microspore. And then this is first cell, and it will increase in size, occupies the entire cavity of archegonium. And the free nuclear division occur, and the free nuclei form, and then central cells, they break down, produces a large cavity. And this cavity, when formed, this stage is called pro-embryo stage. See. Here. This is first divide, and then large cells are formed, and these large cells, then vacuole will come, these cells disintegrate, and the vacuole will form, they are projected towards the outside, and then this structure is called pro-embryo, right? And this pro-embryo further divides into three. See. So wall formation starts from the periphery to the center. It is divided into three: hostorial region, suspensor, and embryonal region. Hostorial is the uppermost, and this is middle, and embryonal is the lowermost. And it results in the formation of pulmule, radicle, and cotyledons, and that is fertilized ovule becomes the seed. Right? Here it is very important to understand that the development of zygote is meroblastic. Why it is meroblastic? Where only the basal cell that develops into the embryo. Upper, they don't take part. So as the other factor is, there is no double fertilization, which we do in the angiosperms. Double fertilization is discovered by Navas-Chin, right? So Navas-Chin discovered